Selamat sejahtera kepada Dr. Rosie and my fellow classmate. Today we'll be presenting about our case study analysis of gas chromatography, or also known as GCs. So we are from Group 5, SES2A. We have 7 group members in total, including Ifan, Shukri, Talika, Emma, Tea, Wen, and Puwa. There will be total of 3 articles used, including determination of decamantine cyclopentacysoloxis in river water and final influence by headspace, gas chromatography, mass, and spectrometers. Another one is determination of polar pharmaceuticals in sewage water in Greece by gas chromatography, mass, and also spectrometers. And lastly, the qualifier was C10 to C13 polychloroalkene and environment sample by high resolution gas chromatography and capture negative ion high-resolution mass spectrometers.
Thank you, Wen. So, my name is Puarasan, son of Anthony Sami. My metric number is E21A0656. I am going to start my part introduction for the case study of gas chromatography. Introduction for the first article. D5 is a chemical in the cyclic volatile methicillocene plus used in personal care products and silicone polymer manufacturing. D5 has been studied using various techniques, including high-performance liquid chromatography and gas chromatography or mass spectrometry. For the second article, pharmaceuticals are discharged into the environment through human waste, and some of the pre-eject medicines and metabolites may avoid removal in sewage treatment process and end up in sewage effluent that reach the aquatic environments. Gas chromatography, mass spectrometric detection following derivatization by diazomethane has been used to detect polar drugs residue in aqueous solutions. For the third article, a technique for measuring C10 to C13 polychloroalkanes in environmental materials is given using high-resolution gas chromatography or electron capturing negative ion high-resolution mass spectrometry in the selected ion monitoring.
Assalamualaikum, my name Muhammad Fandini bin Hazli and my number metric E21A0541, and I will present about gas chromatography analysis technique. Gas chromatography analytical technique for separating and quantifying component if complex mixtures relies on the principle of different partitioning between stationary and mobile phase. Sample injected in gases or liquid form and carried through a column, versatile high separation efficiency and sensitivity. Valuable tool for analyzing complex mixture and trace components. Application: environmental analysis, food and beverage testing, pharmaceutical analysis, forensic science. Next, stationary paste, polymer or solid support coated with a thin film. Mobile paste, inert gas that transports the sample through the column. Elution time, time it takes for a component to elute from the column. Retention time and peak areas used for identification and qualification. Detector measures on concentration of component as a function of time. Chromatogram graphical representation of the sample's composition. Thank you. And I would like to pass to Emma.
Assalamualaikum warahmatullahi wabarakatuh. My name is Irma Nurina Tihani binti Majais. My metric number is E21A0473. In the sampling process, part of this study using Sigma Ardich D5 standard, Moravec Biochemistry D5, acetone N, ultrapure water phosphate solutions, kinesis solutions, headspace glassware, and HP7694 automatic sampler for quantifications. Next is a study using drugs, chlorphenic acid, diclofenac, and others, pesticide-grade acetone, methanol, and sigma PFBBR from Greece for sewage analysis, and finally a study using commercial PCA product and standards analysis. Using 16% and 17 chlorine, isotopically labeled chlorine and myrrh were used, and pesticide test mixture include tozepine, technical chlorine, ultra-scientific, and others. 1, 2, 5, 6, 9, 10 hexachloroquines was synthesized. The study evaluates the extraction efficiency of PCA from Detroit River biota and the recovery efficiency from sodium sulfate. Thank you.
Thank you, Irma, for the good delivery. Now, it's my turn to explain my part, which is sample preparation. Before we start, I would like to introduce myself. My name is Muhammad Shukri Imran bin Muhammad Ishah, ID matric E21A0767. Let's jump to the article number 1. D5 analysis in water samples involve preparing field blanks and calibration standards in a laboratory with reduced background D5. An internal standard 13C5D5 was added to the valve before sealing them in the field. The seal valve underwent irrigation and heating at 80 degrees Celsius for 10 minutes. Gas-tight strings were used to extract 1 ml aliquots for analyzing using the GC split or splitless injector. The method was validated for D5 analysis in distilled water, river water, and wastewater. Quantification was done using headspace GC or MS. For article number 2, sewage samples from high-put areas in Greece were collected every 2 days, stored refrigerated 0 to 4 degrees Celsius, and filtered through a 0.45 micrometer glass fiber filter. A 1 liter portion was mixed with a surrogate standard, acidified, and applied to a preconditioned C18 cartridge for analysis. For article number 3, PCAS in environmental samples were extracted and isolated using modified standard procedures. Frozen fish and zebra mussels were cryogenically ground, mixed with sodium sulfate, and subjected to extraction method. Extraction efficiency was evaluated by spiking sodium sulfate samples with PCA 60 and 13C1 chlorine. The procedures were similar to those used to Detroit River sediment samples. Next, I will pass to Teha and Ika for data and result.
Hello, my name is Nur Fatihah binti Azhar. My matrix number is E21A0645. I will discuss about the data and result for the case study gas chromatography. The case study quantify D5 in water, river water, and sewage effluent using a modified polyethylene glycol column, achieving a detection limit of 6.2 mgl1. It's also investigated potential sewage effluent contamination with the 5 target drugs from 1998 until 1999. Diclofenac was identified as a pharmaceutical residue in all sewage plants. The study highlighted the importance of monitoring ion in commercial polychloroalkane and the need for close relationship between standards and analytes. That's all. Thank you.
Thank you, Shukri. Hi and hello, my name is Nur Fatihah binti Azhar. My matrix number is E21A0645. I will continue to discuss about the data and result for the case study of gas chromatography. A case study quantified D5 in water, river water, and sewage effluent using a modified polyethylene glycol column reaching a detection limits of 6.2 ngl1. The increased recovery and smaller standard deviation were attributed to sewage influent dilution. Experimental sampling on the River Nene at Great Bylin Sewage Treatment Facility in Northampton, UK show a 101% recovery from river water spiked with 192 ngl1. The study also investigated potential sewage effluent contamination with 5 target drugs from 1998 until 1999. That's all from me. I will pass the next discussion to Zalika.
Thank you, Nurul Fatiha. My name, Nurul Zalika Binti Yunus, will continue for third articles that we are carried out from our study, which is the procedure described can be illustrated by estimating the mass of polycarbon alkene in the purge extract on the basis of monitoring the most abundant peak in the analyte and polycarbon alkene 60 primary standard, where therefore use MZ AB stand equal to 0.353. Finally, the concentration of polycarbon alkene in the injected analyte solution can be calculated from the relative volume, nominally 1 microliter by auto injection of injected analyte and primary standard solution by multiplying by R the ratio of the integrated SIM signal of the monitored ion of the internal standard in the primary standard solution to its integrated SIM signal in the analyte solution. Therefore, in the current example, accuracy of the analytical method and choice of standard for quantitative studies. Result for environmental sample recoveries of carbon, chlordine from biota and sediment from the Detroit River are also high, but not for zebra mussel samples from the lake area. This table presents the mean of two levels of polycarbon alkanes detected in this sample. These are therefore lower limits to the polycarbon alkane level actually present. On the assumption of polycarbon alkane recoveries are the same as those of chlordine. We have also estimated in table 5 the polycarbon alkane level that would be present in this sample. Next, I pass our presentation to Liu Hu Wen.
Thank you very much to Thea and Zalika. We'll be wading straight into our conclusion. As throughout many article analysis and research, we have been found out that gas chromatography is a highly effective analytic technique for separating and quantifying a component in complex mixture. The study provides evidence of GS versatility and a wide range of applications in various fields, including environmental analysis, food testing, and pharmaceutical analysis. The article highlights the importance of GC ability to achieve high separation effective and sensitivities, make it a valuable tool for analyzing complex mixture and trace components. Thank you very much for your attention and have a nice day.